Again, thank you so much for attending this session. It's getting late. I'm on the East Coast, and happy to see that there's few participants here interested in learning more about Scipher. I'm going to share my slides real quick, see if it's going to work. And switching to the presentation view here. It should be fine. And I think we can begin. So again, my name is Slava Akmaev. I'm the Chief Technology Officer at Scipher Medicine. I've been in the industry for more than 2 decades, and the last 10 years specifically focused on artificial intelligence and machine learning in life sciences and also healthcare, using molecular data and also data that is more digital in nature, such as patient outcomes data, claims data, other types of data that is relevant to real patient care in healthcare systems. Today, I primarily focus on how we use AI and machine learning at Scipher to develop new products, whether therapeutics or diagnostics. And additionally, how do we utilize experimental data on top of it and combine some of the machine learning technology with experimental biological data. A couple of words about Scipher. We're a small company located in Waltham, Massachusetts. We're a diagnostic company, and we have a CLIA laboratory in Durham, North Carolina. We just recently launched a test in rheumatoid arthritis called Prism Array. And I believe it's a revolutionary test. It's a truly precision medicine test that helps to answer the question of response and non-response for some of the very expensive therapeutics in rheumatoid arthritis. Moving on, a couple of words about our founders. We've been founded a few years ago by Joseph Lascausa, who's Chief of Medicine at Brigham and Women's Hospital here in the Boston area, and Professor Laszlo Barabazi. He's a physicist, and he's been in computational biology for over 2 decades. He's director of complex network research at Northeastern University. Our backers, the company, as I said, been around for a few years, and we have multiple rounds of funding. We have some of the well-known backers in the healthcare industry: Optum Ventures, Khosla Ventures, North Pond Ventures. We're backed by United Health Group as one of the large health insurers in the United States, and a few others. So what is it that we're trying to do? We're primarily focused on autoimmune disorders, and I'll talk more about rheumatoid arthritis today. The idea is that this space of autoimmunity is a little bit of uncharted territories. We know that in oncology, there's quite a bit of development that happened in the last 15 years, where now we understand that patients with tumors respond to therapy primarily depending on the composition of their tumor and tumor-driving mutations. And that's how we begin to select specific targeted therapy for oncology patients. In other diseases, it's not so advanced, and specifically in autoimmunity, there are practically no precision medicine tests, no predictive testing capabilities that would identify and match patients with therapy. So what we're trying to do at Scipher is to find these molecular signatures that would really pinpoint specific therapy, the right therapy for the right patient. Moving on, again, just a couple of words in very general about how we approach dysregulation and autoimmunity. And how does it compare? It's really nice to have a parallel and compare it to oncology, right? We understand now that a lot of the tumorigenesis is driven by mutations in tumors, which is called somatic mutations. And a lot of the development and diagnostic testing sort of evolves around how can we find these somatic mutations, whether in tumor biopsies or potentially in liquid biopsies, the blood testing, and identify the right therapy, diagnose tumors, and sort of improve patient care through that type of approach. And by this time, it's, I don't want to say straightforward, but it's well understood the path for precision medicine in oncology. It's well accepted in the community. It's well accepted in academia, in clinical centers, medical centers, and in research. In autoimmunity, it's a little bit different and maybe in a way more complicated, because we still have in autoimmune disorders rogue cells, right? The cells, immune cells that attack its own host, right? And in a way, it's kind of similar to tumor, where you have uncontrolled growth of cells that come from the same individual. In autoimmunity, you have immune cells that begin to attack the host and cause damage, whether it's joint damage or other types of deficiencies in the human body, right? So how do we identify these cells? Because there's nothing wrong with their DNA. The DNA of the cells is exactly the same as the DNA of every other cell in the body. So instead of looking at DNA, we have to look at other types of measurements and signal in the cellular milieu. And specifically, we'll look at dynamic measurements such as post-DNA molecular data. It can be methylation that is driving gene expression. It can be gene expression itself, looking at RNA expression, measuring it in sort of through this dynamic state. It can be protein expression and protein activity, which is the next stage. So to figure out what is happening in autoimmune diseases, we need to look beyond DNA, and we need to understand a much different set of variables and features and ability to measure them to make an impact in patient care. So what we've done in collaboration with our academic founders and partners is that we developed AI data-driven platform that uses experimental data obtained through thousands and sometimes millions of experimental points, where we put it all together into this network that we call human interactome. And this network represents physical protein-protein interactions that have been verified through biological experimentation. And what we're looking at right now in our development, we can annotate 18,000 proteins in the human cell through this interactome, and then have more than 300,000 connections representing these physical interactions between these 18,000 proteins. This work has been, this technology has been in the works for more than 15 years. It's well published. We have papers in Nature, Science, and other high-profile journals. And the academic drivers and some of the scientific drivers of this technology are helping us to commercialize and lead towards completely novel insight in autoimmune disorders. So what is the unmet need, and what is the question? By certain research, it's been uncovered that about 225 million patients in the world don't respond to their prescription medication. What does it mean? It means that people are prescribed drugs. It can range anywhere from blood thinners all the way to very expensive targeted treatments in oncology or autoimmune disorders. And patients take these medications, they subscribe and they consent for potential side effects, and they practically have no benefit from these treatments. So how do we identify the individuals that are likely to respond? That is the biggest challenge, right? And this is what we're focused on at Scipher Medicine. A bit of a technical aspect of it, everything that we do is represented through networks. What I have here is just a small part of a subnetwork. Let's imagine it's a subnetwork that corresponds to some of the biological signaling or mechanism of action of some of the compounds in rheumatoid arthritis. So then through some of the gene expression data and experimental data that we can obtain in additional experiments and collaborations with academia or medical centers, we can identify biomarkers that may represent patient subgroups that are likely to respond to certain treatments, right? And these subgroups are represented by the ovals that are shaped around the network, right? We have patient subgroup A, may look at patient subgroup B, patient subgroup C, right? All these subgroups and their biomarkers can be overlaid on top of the network. And then what happens is that using AI and machine learning, we can analyze the proximity of known therapeutic targets, whether it's, for example, anti-TNF treatment for rheumatoid arthritis or other types of therapeutics. And by understanding the topology and the relationship between the network and the propagation of the signal from the target to that patient subgroup, we can make decisions by using AI, and actually AI is making decisions for us, whether that patient is a good candidate for that specific treatment. In addition to these sort of high-level approaches where we identify disease modules, we identify network topology, published a number of algorithms and papers that looked at finding therapeutic targets, finding compounds and repurposing opportunities for compounds that are already on the market, identifying drug combinations that may be more potent than a specific therapeutic that we're looking at. Also, as I said, looking at patient individual data in the context of disease module, identifying patient classification, stratification opportunities, looking at precision therapeutics. And then the last thing that I wanted to mention here is probably the most complex challenge in biology and life sciences is to understand biological networks and understand biological pathways in the context of perturbations and in the context of in silico simulations. We published a number of papers on perturbations specifically in the brain and other organs that would able to help the researchers and medical community to elucidate in silico what interventions are more appropriate for specific phenotypes, what interventions can be favorable for people with certain malfunctions and dysregulation in the molecular pathways. So all of these papers are available in the public domain, obviously, and many of these algorithms are available online. But in addition to that, Scipher Medicine have developed their own proprietary algorithms that go even beyond these types of approaches. Now, I wanted to spend a few minutes on our first product that we launched early this year. It's called Prism Array. It is, as I said, first time ever that we have a predictive test in rheumatoid arthritis. And this test really presents the power of personalized medicine in a somewhat prevalent disease, rheumatoid arthritis, in the United States, and allows us to divert patients from unnecessary treatments, save cost of treating patients with therapeutics that don't work, and also improve patient outcomes. So now, in summary, this Prism Array test that is developed by using artificial intelligence and machine learning, and specifically the test itself using a random forest algorithm combining multiple features into one score, allows us to change some of the formulary that is present in rheumatoid arthritis. And for many of you who are probably not that familiar with RA, patients that are about to go on a targeted therapy typically are prescribed a class of medications that are called anti-TNFs. These are medications that inhibit TNF alpha function, and they allow sort of patients to reduce the level of certain cytokines in their circulation, thus sort of ameliorating some of the effects of the overcharged immune system. Not everybody responds, and we'll talk a little bit about more in follow-up slides. But the formulary that are presented by a lot of the insurance companies to the physicians mandate that almost everyone should take an anti-TNF as their first line and go through cycling of anti-TNFs until they go through 2-3 cycles of different anti-TNF treatments and are absolutely, the physician is absolutely convinced that they cannot respond to that medication. So by using Prism Array and diverting people from unnecessary treatments, we can increase the response rate by up to 40%. And you will see that we can reduce overall healthcare costs and avoid expensive and ineffective therapy. So how did we start on this project? It was a really interesting beginning because as orthogonal to many diagnostic companies, we started this journey by talking to some of the biggest insurance companies in the United States. And we asked the question, what is the most critical need for the payers right now in dealing with some of the runaway costs in healthcare? And what is the critical disease that they look at? And what we heard from almost unanimously is that rheumatoid arthritis and anti-TNF medications was some of the most impactful aspect of their healthcare coverage that they experience right now. So we partnered with some of them, and we partnered with the rheumatology communities to work and develop this product called Prism Array using AI and experimental data that we have through our platform. Now, what is the actual need in rheumatoid arthritis? I talked about non-response. Turns out that these anti-TNF treatments are extremely expensive. What you see is $33 billion spent in the United States on anti-TNFs. And it's probably some of the world's largest selling drugs. They're very expensive. A course of treatment can range somewhere in about $30,000 to $40,000 for just a few months. What's interesting here is that 2 out of 3 patients approximately do not adequately respond to that medication. So they may go on a medication, stay on it for say 6-9 months and have no positive effect from the treatment, right? The increase in TNF spend, as you can see in the slide on the left, has increased from 2012 to 2019 by almost 200% with practically zero change in the response rate, right? So we're increasing the use of these medications. We still don't understand which patients are likely to respond, who should get that medication as their first line. And also it's worth mentioning that in rheumatoid arthritis, there are plenty of options as first-line treatments that are alternative treatments to the TNF inhibitors. There are many other really very similarly efficacious compounds that can be used as targeted treatments for these patients. So the Prism Array, we introduced the test recently. It's a 23 biomarker panel. We use 10 single nucleotide polymorphisms, and we have 8 transcript in the panel and 5 clinical features. So as I said, it's combined through a machine learning algorithm called random forest into one scoring function. We have extremely optimistic and enthusiastic adoption of some of the KOLs in the rheumatoid arthritis. They see Prism Array as a foray of precision medicine into autoimmunity and array space in particular. We validated this test in 175 patient population of biologic naive or patients that have not experienced targeted treatments in rheumatoid arthritis from the largest RA registry in the space, the Corona Registry. Maybe not that's such a great name these days, but Corona Registry is really famous in rheumatoid arthritis. They were able to collect information and patient outcomes from tens of thousands of patients. And in addition to that, they were able to run a study to collect biological material, whether it's baseline blood samples or blood samples during treatment from thousands of RA patients. We've partnered with the registry to develop and validate our technology. As you can see on the left, the predictive power of the test is rather high. We have positive predictive value of almost 90%, specificity of almost 87%, and the odds ratio of predicting inadequate response or being inadequate responder in the predicted inadequate response category compared to being inadequate responder in the predicted responder category is 6.6, which is extremely significant. Now, when we look at the clinical utility of Prism Array, if we stratify patients before therapy using Prism Array test, then we can increase response rate for these patients by almost 40%, right? Going from about 30% response rate that we observed in our Corona collaboration to almost 43% if we actually had used Prism Array to stratify these patients to anti-TNF therapy or alternative treatments. So this new paradigm of precision medicine actually allows us to completely shift the structure of and proportions of responders and non-responders. What you see on the left in that pie chart is about 30% of people in the orange are going to respond to anti-TNF, and you have 70% of the population that are not responding to this treatment, right? If we implement Prism Array, and this is what we're doing right now very actively with our academic and commercial partners, then we're changing this entire distribution, right? Now we have about 26% of TNF responders. We have responders to alternative treatments, which is about 16%, all in all, combining to 42% response rate. And then still that we have a pie of the population that is not responding, which requires additional work on other treatment mechanisms in identifying very similar predictive diagnostics so that we can pinpoint exactly which therapeutic they should go on. And then looking at the health economic impact, what you see in the headline is that by using Prism Array, we reduce the net PMPM, which is per member per month cost, by 11 cents, right? And it may not sound much, but if you have if you have a million patient plan, and this is per every member, whether it's person having rheumatoid arthritis or not, this is a significant number that accumulates over the years. And if you look on the right side, overall, for a plan with 1 million members, we're saving about $7.3 million in costs. We're saving $6,000 per tested patient per year, and we're saving $5.9 million in ineffective spend, which is extremely significant. So just to summarize, this ability to predict treatments and ability to actually connect patients with the right treatment will enable us to transition from a rebate-driven perspective on the health insurance side, which means that certain therapeutics are mandated as first line, and insurance companies are encouraged to use those therapeutics by the drug manufacturers by providing certain rebates based on the volume of therapeutics used, right? If there is no precision diagnostics, it's extremely difficult to fight this system. But if we have precision diagnostics, if we have predictive tests, and we can show through validation studies that these tests are working, and we can predict with highly significant, statistically significant power, that we identify patients correctly for the right treatment, then we can transition from the rebate-driven perspective to precision medicine formulary, which how we believe healthcare should be. I'm going to summarize here with our commitment and be happy. We have a few minutes, I think, to answer questions, if there are any.
Yeah, we have until 2:05, like we have 10 more minutes. You can go ahead.
Sorry, I can barely hear you.
We have some more time. We have 10 minutes time. You can go ahead.
Yeah, so I have a question in chat. Igor is asking, is it possible to talk a little bit about particular technological stack behind the platform? Yes, we use everything that we do with our data sets is on Amazon AWS cloud. We use a lot of the analytics tools and some of the tools that are open source for driving machine learning and AI, specifically neural networks and random forest algorithms. And in addition to that, we use some of the open source capabilities to manage our extremely voluminous molecular data.